Good morning all. In the last class we have discussed the characterization of crystal structure using X-ray diffraction techniques. That is one particular class of characterization tool, X-ray diffraction phenomena, X-ray diffractometer. In this class we will be discussing the microstructural characterization and the requirement of the microstructural characterization. So in the case of microstructure characterization we have different scales of microscopes. So we can have the microscopes ranging from the optical microscope, which is the normal microscope, then higher magnification microscope, which is the scanning electron microscope, then the transmission electron microscope. But before moving into the microscopic techniques, we have to prepare the samples. Where in the case of X-ray diffraction, we need not have to make any preparation for the sample. Only we have to make a suitable sample which can be mounted on the diffractometer. But that is not the case of microscope or the microscopic studies. For the case of microscope, depending upon the type of microscopes we are using, we have to make the sample preparations, or we have to do the sample up to the required size. And generally we will be doing the sample preparation for the microscopic studies, where we will start with the sample preparation for the optical microscope. So these are the various steps which is involved for the surface sample preparation for the optical microscope. We normally used to say it as the surface preparation techniques, which starts from selection and cutting of the specimen. Then we will be doing the rough grinding process, then intermediate grinding process, followed by the fine grinding process, and after that we will be doing the polishing process, and finally the etching process. Next we will discuss each one of these in detail. First stage is the selection and cutting of the specimen. We need a specimen of a smaller size. Normally the suitable one is 10 to 15 diameter circular samples, or we can have a square sample or rectangular sample, but it is mandatory that the sample should be conveniently small in size. For small size specimens, which is very small size specimen, we can do bakelite mounting process, where we will be placing our specimen over a bakelite mounting machine, when we will using a suitable substrate which is made up of bakelite to have a sufficient size to do this polishing or the further grinding processes. So if the sample is too small, we have to mold it over a bakelite, so that we can handle it very easily for the further machining process or further grinding process. So first stage is to make a sample or the specimen of the suitable size. Okay, the second stage is rough grinding. We will be using a file or a rotating grinding wheel of larger grain size or grade size, which is driven using a motor for doing the rough grinding of the size. And it is followed by the intermediate grinding, which normally we used to do using the emery paper of having a suitable grid size. So initially we will be using a coarser grid size paper, then it is followed by fine size. So a sequence of grid size it is mentioned, which is 220 grade, 320 grade, 400, 600. As we are increasing the order of these grid, we can have a finer grade or finer grain size of over the not finer size for the emery sheets we will be having. So when we are proceeding from 220 to 600, we will be having a very small sized emery. Okay, so we will be getting more finer sized surface over our specimen. And when we are after the intermediate polishing wheel intermediate grinding, it is followed by the fine grinding process, which is also done by using another set of emery papers. Normally it is named as 1 bar 0, 2 bar 0, 3 bar 0 and 4 bar 0 in the sequence of their size. Okay, the 1 bar 0 will be having the large size, 2 bar 0 followed by 3 bar 0 and 4 bar 0. The 4 bar 0 is the most finest one. So in the 1 bar 0 we will be having comparatively coarser grinding process. 2 bar 0 a size will be much lesser, 3 bar 0 it will be again less, and 4 bar 0 will be the finest one. Okay, now we will be having a comparatively smoother surface over our specimen. Now we have to polish this particular surface by using the polishing technique. The reason for polishing is, even after the fine grinding process of the specimen, we will be having small grooves like or irregularities over the surface, or small scratches. These scratches will be very much aligned, but again it is very undesirable when we are going for the microscopic. So we have to remove these scratches, we have to make it smooth, very smooth of the surface. So this is done using the polishing techniques where the material is removed using the burnishing operation. Okay, so the specimen surface are polished with the help of a rotating cloth pad actually, which is containing suitable polishing medium. So mainly there are two kind of polishing process. First one is alumina polishing, another one is diamond polishing. In certain case we used to do both. Like we will be initially using the alumina polishing technique, then we will be followed by the diamond polishing technique, because diamond polishing can give a very ultra high quality polished surface. Okay, in the case of alumina polishing, the polishing medium is actually an alumina paste. In the case of diamond polishing, it is actually a diamond paste. So what we are doing here is we will be rotating a particular surface, a clothed surface, at a suitable RPM, and we will be pasting this alumina or the diamond polishing liquid over this particular surface. So it will be rotating at a higher RPM, and we will be uh, positioning our particular sample over this rotating surface by a suitable mechanical pressure by using our hand or holding technique. So over a period of time we can see, due to the abrasion between this alumina polishing medium as well as the surface of our specimen, we will be having the material removal, and a longer period of time we can see that we will be having a very smooth surface. So now we have made a surface which is very smooth. Initially we started with a rough grinding process. Initially we started with a sample preparation of suitable size. We have made a cut the sample of a suitable size, which is followed by a rough grinding. After that we done intermediate grinding, and finally we have done the fine grinding process. After fine grinding we have done the polishing process by using either diamond paste or using the alumina paste. Now we have a very fine small scaled uh, specimen with very smooth or polished surface. But now this particular specimen we are placing under our microscope. So before placing into the microscope we have to do a particular process which is called as etching process. This is very important when we are concerning the surface preparation for the microscopic analysis. The reason why we are doing this etching is because one very important requirement of doing the microscopic studies is we have to find the orientation of the grain, we have to measure the size of the grain, we have to know the morphology of the grain. So grains are, we already know, in a polycrystalline material, any material which is composed, polycrystalline material will be composed of lots of grains, and these grains will be meeting each other along the grain boundary. So to differentiate these grains independently, one very good technique is we have to etch the particular sample using a suitable etching medium. When we discuss about the classification of imperfection, we have considered the grain boundaries as a two-dimensional imperfection, or we can consider it as a surface defect. Okay, we already discussed this uh, point. When we are considering a grain boundary, these grain boundaries are the area which have very high chemical reactivity when considered to the inner region or inside that of the grain. So what we used to do is we used to intentionally react this grain boundary with the help of some suitable reagent or some suitable chemicals. These particular chemicals or reagents normally we used to call it as etchants. So etching is a process where we used to attack the grain boundaries to create some chemical reactions at the grain boundary such that we can give a color contrast at the grain boundary when we are observing under a microscope. So if you are giving a chemical attack at the grain boundary, that particular regions where the chemical reactivity has taken or the high surface reactivity has taken place, that particular regions itself have turned to a change in its color, or there will be a color contrast at the grain boundary which can be easily observed at the under the microscope. So we can easily understand how the grains are arranged, how these grains uh, orientation is, or how if we want to measure these grain size we can easily measure if we have an etchant sample or etched sample. So etching is that particular phenomena, and the reagents which we are using for doing this etching process we call it as etchants. Some examples of etchants are shown in the figure. You can see nitrile is one good etchant. Its composition actually consists of 2% nitric acid, 98% methyl alcohol, and it is mainly used for doing this chemical etching or chemical reaction over the ferrous materials, mainly composed of iron in higher concentration. And the time for etching it is normally ranging from 30 seconds to 1 minute. And also certain other etchants are listed over the table. So depending upon the material, we'll be having different etching and we'll be having different etching time also. So we have to optimize before starting our microscopic analysis which kind of etching we have to use for this particular material, and also what is the proper etching time we have to do. Because if we are doing over etching, what will happen is like even inside of the grain will start reacting with this particular etchant. So over a period of time we cannot differentiate the grain as well as the grain boundary. And if we are doing under etching, or we are doing this etching process under a time which is very less than the required time for complete attack of the grain boundary or complete etching of the grain boundary, what will happen is we can have certain regions where we can see this differentiation is the contrast, but it will not be complete. So we cannot completely differentiate the grain boundary, but partially we can differentiate the grain boundary. So etching process or the etching time as well as the concentration of etchant, it is have to be optimized depending upon the type of material we are using. Okay. Next we will discuss the primary characterization of the microscopic technique, or the basic characterization tool which we use for microscopic analysis, which is the optical microscopy. And the device which commonly we use for this optical microscopy, it is the light microscope, or also called as optical microscope. So this light microscope or optical microscope, it is basically used for the microscopic analysis at the basic level. And the major system consists of an optical and illuminating system, and also we should require a surface prepared sample before starting into the microscopic analysis. And how this microscopic analysis takes place, it is basically based on the reflectivity of the light. So the light microscope must be used in a reflecting mode. So the contrast in the image, which the magnified image actually produced as a result of the difference in the reflectivity at the various region of the microstructure. So this particular class of microscope normally we used to for simple microscopic analysis with a magnification normally ranging from 1000 to 2000 maximum. So maximum magnification we can observe using the optical microscope, it is 2000x or 2000 times. And different common modes of operations are 100x, 200x, 400x, 1000x, etc. So we'll be having provision for varying our magnification, and we can achieve the different magnification. These are the basic parts of the optical microscope. We will be having a eyepiece, which is a lens. We'll be having an objective lens. We'll be having provision for placing our sample at the bottom. We'll be having a source of illuminating, or we'll be having a source for providing lighting, which is the light source or mirror or illuminator system. And we will be having a particular provision for varying the position of our eyepiece, or the position of the for varying our focus, which is called as the focus rack and pinion arrangement. Okay, we can vary this focusing arrangement so that we can move our lens to a particular distance. Okay, so the basic characteristic of the optical microscope is the source of light. Source of the beam, it is actually the light beam we'll be using in the reflecting mode. And depending upon the type of the reflectivity from the surface which we are observing, we can analyze the microstructure of the specimen. Okay. So in the next class we will be discussing the electron microscope and the classification of electron microscope. The basic difference of the light microscope or optical microscope from the electron microscope is that in the case of light microscope or optical microscope we will be using a light source, whereas in the case of electron microscope we will be using an electron beam generated using a suitable technique. So the basic classification of electron microscope, scanning electron microscope, the transmission electron microscope, the difference between the two, all these things we will be discussing in the next class. Okay, thank you.